Okay, so let me place this a little bit better. Here we go. Okay, so we have this R2 domain of tau, which is interacting with those tubulins. And what is interesting with that kind of representation is that you can already see here that there is a lysine that is actually forming a salt bridge with the tubulin. And it's a quite important salt bridge because it's maintaining that part of R2 binded to the tubulin. The other part of the interaction, the main part, is over there. This is what we called the strong binding site of the R2 domain. And it's basically, as you can see, buried at the interface between alpha and beta, and the alpha and the beta tubulins. And so on the other side over there, you have some lysins there, but they are only transiently interacting with the alpha tubulin. So it's the weak binding site, and it's the binding site that keeps rearranging itself, which is characteristic of fuzzy interfaces. What else can I tell you? Yeah. So
have you explored that maybe the lysine that are right here maybe could bind to this site right there? Because you mentioned it's a lysine, right?
Yes,
right there, right?
here. Yeah, here it's a lysine.
So is it possible that you know the protein shifts actually?
Not in the time scale that we explore. And the thing is that this is only one domain, one repeat domain, and there are 4 of them that are basically repeating themselves on the surface of the tubulins, and so they all bind basically the same way from what we see now to the tubulin. So this sort of interface, this way of interacting is the same, and I'm not sure that those lysine would be able to interact over there. It's not that favorable. Okay. But the thing is, we're not trying to bind. It's already binded when we run the simulation. So yeah, and so we also investigate the interaction of those C-terminal tails with the R2 domain, and what we found is that these disordered part of the tubulin are actually capable of reaching over R2 and binding over there. And so you have a sort of arc which is wrapping R2 onto the tubulin. Yeah, so it's a sort of,
So what's the time scale of this transition?
So for it to start, it's usually around 100 nanoseconds, and for it to happen, around 25 to 50 nanoseconds for it to completely wrap around. Yeah, it's pretty quick, right? It's pretty quick, yeah. So what we think is that the alpha tubulin is actually capable of maintaining the weak binding region just by binding over. And the whole question that we have now, and this is the reason why we have a bigger system now, is to go with that beta sorry, I've been telling you this is alpha, but this is beta. And the alpha, the question is the alpha tubulin also capable of binding somewhere over there? We don't know because it's just too short, it's got there. So we are running simulations with a larger system to try to answer that question. But it's a work in progress.